Good morning, class. This is Mr. Reynolds, and we want to look more at the eye structures today. We did some of it yesterday, and I thought we should do some of that again because we went through it. It was your first look at it, so you have some things in OneNote where you need to label them, and you're going to need to take some notes on a number of these. Okay, so I want you to be able to refer back to these slides as you need to look at these. I found this in. I thought this was a real good diagram of things that are shown. So I did some fumbling around with it yesterday because there were some things not on here. This is pretty good. The first thing we want to look at is in front of the lens. I'm going green again. In front of the lens, and so the lens is right here. Okay, also called the crystalline lens. This area in front of it is called the anterior cavity. Okay, you see the words chambers here also, but in front of the lens, anterior cavity. Behind the lens, the area back behind the lens, is the posterior cavity, and here it is listed for you also. Okay, so now in the anterior cavity, we have fluid, basically aqueous humor, which is a clear fluid. It's in this front chamber, which is called the anterior chamber in front of the iris. Okay, the colored part of the eye. Okay, is the iris. Now back behind the iris, in front of the lens, but behind the iris. Okay. Is going to be the posterior chamber. All right. So in the anterior cavity, everything in front of the lens, okay, we have the anterior chamber, we have the posterior chamber. Okay. We mentioned yesterday this front part of the eye, the white part of the eye is the sclera, and as it comes around front, very anterior in the eye, it loses its white color and changes clear, and that is the cornea. Okay, we said this outer line. Change my color. My outer line around the cornea is the conjunctiva, and this labels the conjunctiva, this tissue over here on the side. Well, it continues in a real thin line, around the outer part of the cornea. Okay, and this is what can become pink eye. I mentioned yesterday, the second year I was teaching, kids came back from spring break from Florida, and one after the other, came down with pink eye as that bacterial infection was passed around. Okay, and so we said that the formal name is conjunctivitis. That in the posterior cavity, we said it is mainly a clear gel. Vitreous humor is a clear gel. And like I said, we need all these structures clear, like a window, like the light in the room and the window, just for light rays, so they can pass through that. They can pass through the clear conjunctiva, the clear cornea, the clear fluid, through the little opening in the lens, the lens is pretty much clear anyway. Okay. And then the light rays get bent, and it heads over. Most of the time, as we're looking straight, it hits on a structure. I'm going to change this to yellow. This structure is actually it's yellowish when we look at this, and I'm hoping that a dissection turns out on Friday. They're going to film that okay, and you'll see that this is a dark black lining of the retina inside, and that there is a spot, there's a yellowish spot, okay, and at the very, very center of that spot, at the very center of the spot is what's called the fovea centralis, okay, and this enlarges it up here for you. It is a little bit of an indentation that we see, okay, and we said there's tons of cones in here. And here's a cute shaped little cone there, like an ice cream cone, okay? Others are more of a rod shape, all right? And like I said, we'll study those a little bit more, and we said those are for black and white vision, and cones are for color vision, all right? But it takes a lot of energy making these things, especially these cones. We need a lot of light to be able to see. So at night, in the evening and nighttime, there's not as much light, so they wouldn't work anyway. If we had cones all along here, it just would not work, okay? So because there's not enough light. So most of the cones are centered right here in the fovea centralis. Okay, the optic nerve comes into the back of the eyeball, and I didn't say this one yesterday, but where this is, there's no photoreceptor cell. There's no rods or cones here in this area. Okay, there's none. So if the light rays hit on here, if the light rays hit on the optic disc where the optic nerve comes in, you don't see anything. And so we said the name is also called a blind spot. So if the light rays hit on that, you don't see anything. Okay, these three layers, the retina, the choroid coat, and the sclera. Okay, three layers of what's called a tunic, are the layers of the cell. Inner tunic, outer tunic, middle tunic. Okay, and you need to, when someone's talking about the layers of the eye, well, are they talking from the outside in, or are they talking about from the inside out? If they're talking about the first layer, are they talking about the one on the outside? They're talking about the white of the eye, the sclera, and then the choroid coat is the second one, and the retina is the third one, or are they going opposite direction? Do they mean from inside out one? So just clarify that as you're doing it. We'll talk from outside going in. We said the white of the eye is the sclera. Okay, that's the first outermost layer. Then a colored layer is the choroid coat. Okay, we say as it comes around here in the anterior portion of the eye, it makes up this colored ciliary body, the colored part of the iris. Okay, and then we said these suspensory ligaments hook onto it. Okay, here's the word up here in the top, suspensory ligaments. Okay. We said this opening, the pupil. The pupil is not really a structure, it's this opening between the different parts of the iris. We said in the morning, it's open really, really wide, and when someone turns the light on, it just comes flooding into you, and it's going to hurt. All right, until about 20 seconds, the iris has a chance to constrict. It's called the pupillary reflex. What we want to look at more is the retina. Let me describe this a little bit more. Like you'll see it cleared in the dissection. There aren't very many fibers that attach the retina onto the choroid coat, and this can easily become detached. There's a problem that's called, some people have, and you've heard this before, a detached retina. Okay, a detached retina. I was in the office one day, and a student who wasn't going to school there anymore, he had stopped by the office, and it turns out that he had just gotten out of eye surgery about 3 weeks or a month before that. He looked pretty normal, and I said, "What happened?" He said, "Well, my buddies and I were playing paintball, and I just I got sweaty, couldn't see. It was kind of stupid of me, but I took my safety goggles off, and right then I got hit in the side of the head with a paintball, and his eye was hanging out of his orbit, and so they had to surgically put that back in. And the eye is not super solid in that orbit." So anyway, I just thought that was wild. If someone gets a blow to the eye, they can get a detached retina. Even if the eye looks normal, is in place, they can do that. An analogy that I use for this, and pay attention here, if you have a trash can and you have one of the plastic garbage bags sitting in there, okay, if you fill that with water, okay, or if we filled it with gel, cleared with gel, fill it with water, okay, and that's going to shove the plastic bag onto the right up snug against the lining of the against the trash can. Now, if there's a little cut in that bag, then water's going to slowly seep out of it and seep behind there, and the bag is going to come sloughing off of there. Okay, that's basically what a detached retina is. There aren't any there's almost no fibers attaching the retina, holding it onto the choroid coat, so it can pretty easily become detached. Okay, let's move on to the next one. Got a few more slides. And that first one was the big one. That had most of the major stuff on there, and there's some other things I wanted you to look at here. Now this thing is, what we're looking at here is the retina. Okay, that inner lining that we just looked at. And what's wild about that, here's rods, and here's some cones down here, and like they're named because they're shapes. And the rods have more of a rod-like shape on it. Cones more of an ice cream cone shape. Okay, the light rays come in through the top. The light rays come in through the eye, through the top. They go through these nerve cells up here in the top portion of it. They'll go through that, and they stimulate the cones and the rods when they get down here. Okay, and if a cone gets stimulated, let's say this cone right here, let's say it gets stimulated with light rays coming in, it sends an impulse up to this bipolar neuron that tracks it up to other nerves that track it up to the brain. Okay, so this is off to the brain. Okay, now each cone, let's go back down to this one that I drew. Each cone has one nerve cell that it sends an impulse to, okay, and it goes up to the brain. This cone gets stimulated, it sends an impulse through this nerve up to the brain. This cone gets stimulated, one nerve carries it up to the brain, okay? So it's basically, cones are a 1-to-1 ratio, extremely specific vision, okay? And all of this, most of these cones are, we said, in the fovea centralis, in the macula lutea. Okay, the rods, if this rod is stimulated, it sends impulse to this neuron, or it could be this one sends one to it. Okay. Oh, and over here I have 1, 2, 3 different rods stimulating the same neuron that sends impulse on up to the brain. So when the brain gets this message, well, it could have been from this one or that one or that one. And I've seen some with 5, 6, 7, 8 rods attached to it. So the rods, it's not as specific vision. And notice with us, well, we see in color, so we're using our cones a whole lot. It's extremely specific vision, 1-to-1 ratio on it. Okay, next slide. This is reviewing from yesterday's quick review here. This is my left eye, this is my right eye. Anything that's coming into the right eye is tracking up to the brain and is registering over here in the left hemisphere of the cerebrum. Anything coming into the left eye is tracking up to the brain over into the right hemisphere of the cerebrum. And this structure right here, where these nerves end up crossing a whole lot, is going to be called the optic chiasma. So we got a number of things happening here. You actually are seeing 2 images coming in, one registered here, one registered over on the right side, but you see it as one. Your brain does that. So the answer is going to be in these billions of cells right in here in this optic chiasma. Okay. Next slide goes along with that also. So as we see something like this capital F, right side up, as it comes in through our lens and the rays get bent and they hit a focal point right here and they go past that, oh, now that object is upside down. And those impulses hit the retina. This is the retina here with the rods and cones we just saw. And it's going to track it up to the brain through the optic nerve, okay? Up in that optic chiasma, up in your brain, that's where this image is inverted back around. It's turned right side up. Okay. Next slide. This is the last one. I said this yesterday. I wanted to say this again. The accommodation is the process, and I'll see if we can do this. See if we can do this in the lab tomorrow, or on Friday when you see this next. We said there's interplay between there's an inverse relationship between the tension on the ciliary muscles. I'm coloring these in blue, okay, and between the suspensory ligaments. I'm going to write this word on here again, okay, because this is these two, the muscle and the ligaments, specifically suspensory ligaments, helping to suspend the lens, suspensory ligaments. If this muscle is relaxed, the ligaments are tighter, okay? Down in this one, if the muscles are contracted and tighter, the ligaments are looser. And if it's looser, less tension on the lens takes more of its shape. And notice it actually describes it as bulging some. Okay, it takes more of its natural or ovoid shape is what I was taught. Okay, but you know, we said with this muscle, with the muscle up here relaxed, putting some more tension, ligaments tense, lens flattens out. We can do that all day long because the muscles are relaxed. Down here with the muscles tense, with the muscles contracted, ligaments relax, lens takes more of its normal shape, and this is for close-up vision. Well, like reading, we can only do that so much because these muscles are going to fatigue out. But this whole process is called accommodation, okay? You can kind of do this if you have a piece, something in front of you, put your finger down by a letter, okay? So that your eye can go directly to it. You're going to look off at something way in the distance, and just kind of relax. Look at that for about 5 seconds. 1, 2, 3. Look down quick. There's less than a second, maybe less than half a second. It was just some haziness when you look at that little letter, as it takes time for these muscles to, tighten up, and for this bottom diagram to occur. Muscles tighten, ligaments relax some, lens takes more of a bulging shape, and we can focus on the object closer up. Okay, if you want to do that again, that's something called the accommodation reflex as we're doing that. Okay, and